Here we're just going to do a quick review of some terms which you may have already heard about. The first term is the term genotype, and the other term is the term phenotype. So a genotype is a description or a labeling of what versions of a gene a specific organism carries. So basically it's saying what alleles does this individual organism have for any particular gene. So to keep continuing with the example of the petal colors, we said that there was a capital P version of the gene which created a purple protein, and then a lowercase p version of the gene which created a white petal, a white protein. So what the genotype tells you is any individual, do they have two big P's, the ability to make the purple protein twice over? Do they have one of each, or do they have two copies of the version of the gene that makes the white protein? That's the genotype. So whenever any question is asking you to write down a genotype, it's always going to be two letters if it's a diploid organism. So genotypes are going to look like this or like this. That's a genotype. Tells you the genes or the alleles, the versions of the genes that any individual carries. Now, a phenotype, this is easy to remember because it begins with PH, and I like to think of it as like the kind of physical characteristics. So the phenotype is the physical characteristics. And a phenotype is going to be descriptive. So for example, purple colored petals or white colored petals. Or me, my phenotype could be 5'7", blue eyes, brown hair. It's a description of how the thing is looking or behaving or functioning. So it's not necessarily telling you specifically about the alleles or the genes. It's like, what does that individual or organism look like? So on this chart, we can see that the phenotypes of these flowers, we've got 3 purple flowers. So these flowers all have the same phenotype, and this has a different phenotype, it's white. But if you dig into the genetics, you might not be able to tell what the genotype is from these purple flower plants. This 1, 2, 3, they all look identical. So their phenotypes are identical, and if you didn't know the background of how they'd been bred or what their parents were, you wouldn't be able to necessarily tell what their genotype was. You can tell their phenotype, it's purple, but you can't necessarily tell their genotype if you only have the information on the right side of the screen. Now, if we look on the left side, it's actually telling you the genotype or what alleles these individual plants have. So this one is a capital P, this one's a uppercase P, capital P, lowercase p, this one's uppercase, lowercase. You can see here that these, although they have different genotypes, they have the same phenotype. That's one thing to be aware of. And there's also some terminology here you probably already heard of. So the first terms are homozygous and heterozygous. All right, so homo means the same. So if both your alleles are the same, you are homozygous. So this is a homozygous situation, and this is a homozygous situation because this is both the same, big P, and this is both the same, little p, little p. Heterozygous means that the two alleles that you have are different. And here you can see big P, little p, they're not the same, they're different alleles. Now, that's all well and good, homozygous, heterozygous. But because both this and this are both homozygous, we have to have another term to separate one from the other. And so we have this additional phrase that we use, which is dominant and recessive. Now, these terms were first used by Mendel, and they meant something back then, back when Mendel was describing what's going on. Unfortunately, these terms of dominant and recessive are not as helpful anymore as now we know more about what's actually going on in the background, in the genetics and the molecules that are actually going on. These terms are a little misleading. Now, all that dominant means, all right, is that In the heterozygous condition, so this is the heterozygous condition, big P, little p, right? The dominant allele is the one that you see in the phenotype. So what I'm saying here is that this plant here is big P, little p, right? But you see purple. So we're going to say that the purple color is the dominant allele. All right. Now, does that mean that, and the only reason we're calling it dominant is that you can see it in the heterozygous individual. It's the purple that you see, not the white. All right. Now, does that mean that purple is better, stronger, healthier, correct? No, it doesn't mean any of those things. All it means is that, or does it mean that purple is, because we think of dominant as being like powerful and pushing down and controlling and turning off and in charge of, right? Those words are just our projections onto this. The purple gene is not stronger than, more powerful than, turning off, pushing down, getting rid of in any way the white gene. The white gene is also there and it's totally fine. It's just that our eyes can't pick it up. So the idea that we would call purple dominant is really more of an idea about our eyes. What can our eyes pick up here? It doesn't mean that it's pushing down the white one or turning it off or overpowering it or anything like that. And so it's sort of one of the weaknesses of these terms that we've inherited from Mendel, because to him it probably did seem that purple was stronger and pushing out white. But now we know the proteins behind all this. We know it's not doing that. But we're stuck with these terms and they're kind of historical, so we've kept them. But all that dominant means is that it's the one that we actually physically can detect with our eyeballs in the heterozygous condition. And so we call big P, or the two uppercase, the dominant situation, homozygous dominant. And then the allele that you don't see in the heterozygous case, which would be the white one, we call that recessive, or Mendel called it recessive. And so we have these terms now. Homozygous dominant means you have two of the same and they're the dominant allele, or homozygous recessive, which means you have two of the same and they're the recessive alleles. And then you have the third term, which is heterozygous, which means you have two different ones. And you never, ever, ever say heterozygous dominant or heterozygous recessive. That does not exist. That's a term that doesn't exist, because heterozygous just means that you have one of both already. So you can't be heterozygous dominant or recessive. It doesn't make sense. So never write heterozygous dominant or never write heterozygous recessive. That's the wrong thing. You just write heterozygous on its own, because it means you have one dominant, one recessive. But that's how these terms work together. You can also see here that in the case of the white phenotype, you know the genotype. The only way you can be white here is if you have two little p's or you're a homozygous recessive. So sometimes you can tell a genotype from a phenotype, for example, in the white case. But in the purple flowers case, I can't tell. If I only saw the flower and I didn't know the parentage, I couldn't tell by looking at this flower whether they're going to be big P or big P, little p. There's no way to tell by looking at the phenotype in that case. All right, I hope that helped clear up some of those terms, and we'll move on.